Analysis of Biology Question 2, Konkoor 2025. Griffith's experiments. Direct hit goal, understanding the examiner's logic. Iran second turn. Hello everyone. In this video, we are going to analyze question 2 of the Biology Konkoor 1404, a question based on Griffith's experiments, which includes a very common conceptual trap. Question 2. Which of the following statements about Griffith's experiments or their results is incorrect? A. They ended the long-term debates and research about the nature of genetic material. B. He discovered that a heritable trait can be transferred from one living cell to another type of cell. C. In one of his experiments, he observed that many non-capsulate bacteria became capsulate. D. In one of the experiments, live capsulate bacteria were observed in locations other than the blood of dead mice. The question asks, which statement about Griffith's experiments or their results is incorrect? Pushpin key point. Since the question asks for the incorrect option, we must carefully compare each choice with the textbook and the logic of the experiment. Griffith worked with two types of Streptococcus pneumoniae bacteria, smooth S bacteria, which have a capsule and are pathogenic. Rough R bacteria, which lack a capsule and are non-pathogenic. He observed that, when heat-killed S bacteria were mixed with live R bacteria, the R bacteria became pathogenic. Pushpin. This phenomenon is known as transformation. Small blue diamond option A. It ended the long-term debate about the nature of genetic material. Crossmark incorrect. This is the correct answer to the question. White heavy checkmark explanation. Griffith's experiment did not determine what genetic material is. It only showed that a heritable factor could be transferred. Later scientists, such as Avery and Hershey and Chase, completed this research. Small blue diamond option B. He discovered that a heritable trait can be transferred from one living cell to another. White heavy checkmark correct. Because the pathogenic trait was transferred between bacteria. Small blue diamond option C. In one experiment, he observed that many non-capsulate bacteria became capsulate. White heavy checkmark correct. This is a direct result of genetic transformation. Small blue diamond option D. In one experiment, live capsulate bacteria were found in places other than the blood of dead mice. White heavy checkmark correct. This indicates that the bacteria survived and multiplied in the mouse body. The key message of this question is very important. Griffith did not identify genetic material, he only showed its transfer. Any option claiming that the problem was completely solved should immediately raise suspicion. If you want to think like a biology examiner, make sure to follow this channel.